So hey, it's Riffy. Welcome to your absolute positive first section of notes that you will get to experience in this beautiful, beautiful format of Riffy's flipped video notes. So let's just dive right into this stuff. We have one thing in our crime scene that we have to identify whose it is, and it's the blood. We can't do the hair, we've discussed that because there's no follicle. Fingerprints don't leave DNA. We can't do the vomit because the DNA gets degraded because of the acidic environment in the stomach. So really and truly, we need to analyze the DNA in the blood. How does that happen? Well, DNA is inside all of our cells, but not quite every single cell in the human body. You've been told up to this time frame now that all the cells have a nucleus, and inside that nucleus is DNA, and the DNA determines the characteristics. But really and truly, they've been lying to you. Not every single cell in your body actually has DNA inside of it, particularly red blood cells. Red blood cells don't have a nucleus, they don't divide, they don't replicate, they are actually made from other pre-existing cells, so they're an exception to that rule. But pretty much everything else has DNA inside of it. Therefore, any type of human cell or tissue that's left at a crime scene can essentially be used to identify who was at the crime scene. So now our main question is, who was at the crime scene and whose blood was it? Is Alex going to be implicated in this, or is this blood actually Anna Garcia's? Before you can do any of that stuff, you got to understand what DNA actually is composed of. The composition of DNA is actually what leads us to be able to identify who was at a crime scene, what happened at a crime scene. Is it his blood? Is it her blood? You know, all of those things. So let's dive into actually the structure of DNA so you can understand that, so we can move forward in the class to identifying who done it or who didn't. So let's get into actually the background information of DNA. What you need to understand is in living tissue or living compounds or living organisms, however you want to group it, living things, there's going to be four main groups of biological compounds that pretty much the entire structure of the body can be divided into. Those four main compounds are going to be your carbohydrates, they're going to be your lipids, they're going to be your proteins, and boom, the fourth category that DNA belongs to, which is going to be called nucleic acids. DNA stands for deoxyribonucleic acid, hence why it's part of the nucleic acid group. The other type of nucleic acid that's going to be found in the human body is RNA, but we're going to get into that later on in the class in a little process called protein synthesis. But let's get some more background information about DNA. It belongs, like I said, to one of the four main biological macromolecule compound groups called nucleic acids. One of the defining characteristics of nucleic acids, besides the fact that it's extremely complex, is that it's only made from the elements of carbon, hydrogen, oxygen, nitrogen, and phosphorus. The defining element in this category is going to be phosphorus. If you see phosphorus in any diagram or picture of a molecular compound and somebody's asking you, hey, I need you to tell me which of the four main groups it's going to be, this compound is going to be part of, you instantly look for phosphorus. If you see phosphorus, don't write anything else down except for DNA or RNA. Boom, nucleic acids. Now, DNA is inside of our cells, and like I said in that introductory slide, it's not in all of our cells, but in pretty much all of the cells in our body, except for red blood cells, and maybe a few others. We don't need to mention those. But DNA inside our cells, inside our nucleus, actually determines our inherited characteristics. This is an extremely important statement that you see here, and the fact that it says our inherited characteristics. What I mean by inherited characteristics is it takes the half DNA that you got from your mom, the half DNA that you got from your dad. When there was egg and sperm, they come together, boom, they fertilize, and what happens is the sperm kind of dissolves or disintegrates or disappears, but the DNA that was held inside of that sperm cell gets incorporated inside the egg cell. So you literally are starting with two half sets of DNA that combine to form a full set, and from there a bunch of chemical reactions determine every single individual cell inside your entire human body. That's why it's called the blueprint to make all cells, because literally life is starting with an egg and a sperm fertilizing, and nothing really in there except for the DNA is going to determine what comes out in the end result as a human being. So how does all of this occur? In a very, very, very basic format, you're going to notice that this occurs through a process of biological reactions or steps, but it's going to always start with DNA. So I'm going to go ahead and write this out real quick. If it starts with DNA inside the cells is going to be used to create a compound called RNA, which is still within the nucleic acid group. That RNA then, once it's created, is going to leave the nucleus of the cell and it's going to go to the cytoplasm, and it's going to be used in translation, which is part of protein synthesis, to actually create a series of amino acids. That series of amino acids is then going to form a compound called a protein. Proteins are the most abundant and diverse compound in the entire human body, largely because they're used in chemical reactions to determine all of our traits. A trait is a characteristic. You need to understand, however, that traits are not just your physical characteristics. Your traits are actually going to be anything that is anatomical, so like this beautiful, balding, brown-eyed, bearded face that you see in front of me is determined by my DNA and all of this series of chemical and biological steps that ends up with my traits that you see here. But you also need to realize that all of this occurs and determines your physiological traits. This is how your body works. So your DNA and all of these chemical reactions that occur determine things like your ability to break down lactose from milk. So if you're a lactose intolerant person, blame the series of chemical reactions here that's determining that trait. We can get super extreme with it. For example, Down syndrome can be caused by an extra 21st chromosome. Well, that's throwing more DNA into the mix, which then causes all of these things to change and ends up changing the traits. So that's why we say DNA holds the blueprint for creating every cell in the body, because literally the entire human body only starts as 2 half sets of DNA. So let me get back through these notes real quick, since I just used it to draw. And we're going to get into now what are the actual individual components of DNA. Before we can understand this, you need to understand that DNA, being a major macromolecule or a large biological compound, essentially is called a polymer. Polymers are very large compounds that are made from smaller components called monomers. You take several monomers, you put them together repeatedly over and over again, and you end up with a polymer. Over here on the left hand side, I got a video playing real quick. And in that video, you're going to notice that it starts out with a really complex DNA molecule, and then it's going to go ahead and take itself and it's going to split or flatten out first. And in that flattening out, you'll notice that there's a repeating pattern to the entire DNA molecule. If you break that down into the individual pieces, what you're then finding yourself is you're finding yourself discovering the monomers. Monomers are the repeating units that make up polymers. So if we let this video fly just a little bit longer, you'll notice that it also kind of does a little tricky thing, and boom, it's going to split in half. When that splits in half, the reason why I have this here is I want you to realize that yes, DNA is a polymer, but it's actually a polymer made from 2 long-stranded polymers. So looking at this molecule, this itself is one polymer and this is the other. And DNA is comprised of 2 polymers bonded together in the middle. So again, rewinding, DNA is a polymer comprised of repeating units called monomers, and that particular monomer of DNA is called a nucleotide. Each of those biological compounds that we talked about earlier, the 4 main categories, has their own monomer. We're not going to get into those right now. We're just going to concentrate on DNA. So this other video that I have over here is actually going to show DNA coming back together after being split apart. Back into its complex form. Even in that complex form, you can see all the individual monomers built together to comprise that one DNA molecule. So now we're going to study the individual structure of the nucleotide, or the monomer or building block of DNA. The reason we're going to study the structure of a nucleotide is because really and truly the sequence of nucleotides is what determines this beautiful mug you see right here. All the different parts of a nucleotide really come down to only 3 items. You have what's called a phosphate group. That phosphate group bonds itself to a deoxyribose sugar. That deoxyribose sugar bonds itself to what's called a nitrogenous base. Those 3 things alone are a nucleotide. Now this diagram is really simplistic, but it has to be in order for us to build the steps up to understanding the more complex. So a single nucleotide is simply made from these 3 structures: a phosphate group, a sugar, and a nitrogenous base. The phosphate group and the sugar are going to work together to make up the backbone of DNA, while all the nitrogenous bases in the middle are going to make up like the rungs of the ladder, so they make up the center portion. And what happens, remember we said monomers are the building blocks and they repeat themselves over and over and over again in the compound to form a polymer. Well, you will see that they bond at the phosphate and sugar backbone to form one side chain or polymer. So really and truly, as it forms, you're going to call this one of the polymers right here. Boom. DNA being comprised of 2 polymers, you'll have the monomers repeating themselves over and over again on the other side, but notice one particular characteristic of it: it's in the opposite direction. So these sugars are pointed upwards according to the oxygen, these sugars are pointed downward. If you watch the Bozeman biology one, he talks about 5' versus 3' and the different ends of the DNA molecule based on the types of strands. That's something for a genetics class or our 3rd level of biomedical science. As far as it's concerned right now, you just need to know that there are 2 polymers. One goes in one direction, the other goes in the other. Each of those polymers are comprised of nucleotides, which is the monomer, and each monomer is composed of a phosphate, a deoxyribose sugar, and a nitrogenous base. However, you should be questioning right now, what's going on here? You told me these 2 things are come together and are bonded. Well, if you notice right here down the middle, there's something missing. You're missing the connector that brings both polymers together to create the entire DNA structure. Well, that missing component is actually going to be called the hydrogen bond. Hydrogen bonds are a very weak bond that hold together each strand or polymer in that DNA. It actually needs to be a weak bond. Not getting into why, but it has to do with splitting the molecule, creating RNA, DNA replication, proteins. That whole sequence of actions that we went through requires hydrogen bonds to be a weak bond. Now, I know you're thinking, wow, that was a really simplistic diagram. I can memorize that for the test. I know what DNA is made from. Boom, I'm a geneticist. I'm the smartest person ever. But unfortunately, you had to know it had to get a little bit more complicated. And that more complicated version, the reason why I'm throwing that in here is because this is literally the model you're going to try to build in class. Your model is complicated. It actually represents each of the individual atoms involved in the phosphate group, involved in the deoxyribose sugar, and involved in each of the nitrogenous bases. So this complex version that you're seeing right here, you're going to actually build in class with the model kits. Let's get into the nitrogenous bases, and here is why. There are a total of 4 different nitrogenous bases in DNA, and if you throw RNA back into that mix, that makes a total of, in the entire nucleic acid category, a total of 5 nitrogenous bases. But let's just concentrate for now on the nitrogenous bases within DNA. There's 4 different ones. You have one that's called adenine. Everybody say adenine. Excellent. You have the other one, which is called guanine. I want you to take a good stare at these 2 right now and look at what the common characteristic is between them, because we're going to revisit that here in a second. So you have adenine here, A-D-E-N-I-N-E, and you have guanine right here. Look at them. Notice the similarities. The other 2 that you have are called cytosine. So this one's going to be cytosine right here. You like my handwriting? And then the last one is going to be called thymine. Keep in mind, what's the common characteristic? Oh, you've already identified it. Well, it's the fact that they have a structural component similarity between the two. If you'll notice, the two over here on the left-hand side that are thymine and cytosine only have what's called one ring. That one ring is this thing right here and this right here. That common characteristic alone is what separates them from the other two, which have two rings. 1, 2, 1, 2. What that allows us to do is within those nitrogenous bases, we can actually split them into two separate groups. Those two separate groups are going to be called purines and pyrimidines. The purines are the ones that have two rings. So together, these two things form a category called purines. That would be the adenine and the guanine comprise the purine group. The other group that is composed of single rings are going to be called the pyrimidines. The pyrimidines, being cytosine and thymine, only have one ring, whereas the purines have two rings. Yes, you're going to have to know that for your test, and yes, you're going to have to be able to identify them individually. So how are we going to do that? I'm going to show you. So if I asked you to look at this diagram here, which is essentially the exact same picture we had before, but now I'm representing it slightly different, you will notice that guanine and cytosine, which are the two down here, so you have your G and you have your C, and your adenine and your thymine here, you will notice that there's a characteristic that even though these two are the purines on this side and these are the pyrimidines on this side, you'll notice that even though they have different ring structures, one ring here, two rings here, that there is a commonality between them. That commonality is the number of hydrogen bonds. So adenine and thymine form two hydrogen bonds, and guanine and cytosine form three hydrogen bonds. What that is called complementary base pairing. The rule is adenine will always bond with thymine, and guanine will always bond with cytosine. And they bond based on these three hydrogen bonds and the two hydrogen bonds here. That is called complementary base pairing. What does that allow us to do? Well, complementary base pairing actually allows us, if we were giving one entire strand of DNA, we can actually predict the complementary strand based on that characteristic. So if I gave you this one side, this one strand of DNA, not one strand, I'm sorry, one half of the DNA molecule, I would like to know if you can give me the other half. So that's one polymer of the DNA molecule. What's the other half that be bonded with the hydrogen bond? Well, based on our rules, A always bonds with T, C always bonds with G, G always bonds with the C, and A always bonds with T. Back and forth, back and forth. This bonding characteristic right here is going to give us the actual other half of the DNA molecule. Keeping in mind, this one goes upward and the other one goes downward. A and T bond with two hydrogen bonds. C and G, even though I kind of messed up my diagram a little bit, this should be bonded with three hydrogen bonds right there. So, complementary base pairing. A purine always bonds with the pyrimidine. A always bonds with T. C always bonds with G. And the bonding is based off of the number of hydrogen bonds that can form between the two. That is called complementary base pairing. What does complementary base pairing lead us to? Well, it leads us to complementary strands. Keep in mind again, we're going back to the same topic. DNA is a double polymer basically. It is comprised of two polymer strands that are bonded together in the middle. If you know one strand, you can know the other based on complementary base pairing. A always bonds with T, C always bonds with G. AT, CG, TA, AT, CG. So if I only gave you half of the strand, you should be able to give me the other half of the strand. But this genius base pairing and complementary base pairing and complementary strands actually gives us this fancy thing called Chargaff's rule. Everybody say Chargaff. Nice. Chargaff's rule essentially means if you know the quantity of one of the nitrogenous bases, you know the exact quantity of the other nitrogenous base. Here's what I mean. Let's say, for instance, I'm examining a DNA strand and I find that it has 42 adenines. It also has 68 guanines. And it has 18 cytosines. And I don't know. Let's go with 24 thymines. Let's say that's just this strand right here. If I asked you how many of every nucleotide is on the complementary strand, could you tell me? The answer has to be yes. Here's why. If you have 42 A's on this strand right here, complementary base pairing tells us that every A on this strand is going to bond with a T on the complementary strand. That means for every A on this strand, there's a T on this strand, meaning that there is in fact 42 T's on that opposite strand. If we're talking about the, if I said that there were 18 guanines, I don't know why I have trouble writing an 8, on this side right here, that would have to mean on the opposite side there's 18 cytosines. That's called complementary strands in Chargaff's rule. Chargaff's rule basically means the ratios of nucleotides are equal on opposite complementary strands. There's always going to be the same amount of A's as there are T's on opposite strands, and the same amount of C's as there are G's on opposite strands. Do you like my half head that you've been seeing the whole time? What does this lead us to? This leads us to DNA and the double helix. DNA, after all of these monomers get put together over and over and over and over and over again, you'll notice that the natural combination in a way of each of the sugars, the phosphates, and the nitrogenous bases come together is it naturally causes DNA to form this twisted double helix structure. It's called like a twisted ladder. I'm not going to tell you why it naturally forms the double helix. I want you to figure that out based on your molecule that you're going to make in class. So here's some hints as to why DNA forms a double helix when all of those monomers are put together. You need to pay attention, one, to the size of each of the nucleotides and the size of the complementary base pair that it's going to bond with. They could either be the same size or different sizes. But if they were all the same size, I want you to pay attention to what would happen. I also want you to pay attention to the size of the base pairs if you tried to bind them incorrectly. What would that do to the structure of DNA? The third thing that's going to give you a hint as to why DNA forms a double helix is when the nucleotides actually complement each other, what does that cause the sugars and phosphates to do on the outside rings or on the outside rungs of the ladder? So hopefully this video wasn't too long for you. Hopefully it actually makes sense. You know, it's like 11:45 at night and I'm just now wrapping this up, so my brain has kind of stopped working. Hopefully this clears some of the stuff up about DNA, but I would love to help you out if you need anything. Best of luck.